Hi there, folks. Welcome to Lesson 6 of Unit 1 of the National 5 Biology course. Today we're going to be focusing on Topic 6, which is all about respiration. Probably one of the topics that students do find the trickiest in Unit 1. So we're going to go through everything and hopefully you'll feel a bit more confident and comfortable with it by the end of this lesson. So the first thing to say about respiration is a process which happens in our cells and is controlled by enzymes. Now, 2 lessons ago, we learned about enzymes and how they are biological catalysts that speed up cellular activities. This is one of those cellular activities that those enzymes speed up. So, the first thing to remember, and this is a question that does come up from time to time, is that respiration is controlled by enzymes. And you're often asked things like, if the temperature increases suddenly, how does this affect the rate of respiration? Well, if the temperature increases, that will denature the enzyme. And because respiration is controlled by enzymes, that would then obviously slow down this process. So that's worth remembering. That's a common exam question that can come up. So, what does respiration actually involve? Well, it involves the breakdown of a substrate. Remember, enzymes need a substrate. It involves the substrate, glucose, being broken down. Okay. Now, glucose, like many foods, contains chemical energy. So, we need that chemical energy to be released for a series of reactions that allow us to make an energy molecule within the body. The chemical energy stored by glucose must be released by all cells through a series of enzyme controlled reactions. And all of those reactions collectively are known as respiration. Now, the energy that's released from this breakdown of glucose is used to generate our energy currency within the cell, the molecule that gives us the energy to do many of our most important functions. And that molecule is called ATP. Now, ATP stands for adenosine triphosphate. Okay, so you can see here we've got a adenosine molecule here, and we've got 1, 2, 3 phosphates here. Adenosine triphosphate, ATP. Now, that's important to remember because when we use up ATP, when we use ATP for energy within the cells, what happens is the third of those phosphates breaks off. Okay, the reaction breaks off the third of those phosphates, and the energy that's released from that is used by our cells for various activities that we'll talk about in a minute. However, the great thing about this mechanism is that simply by adding that phosphate back on, by building it up again, okay, so adding the third phosphate back on, we can recreate our adenosine triphosphate, or ATP. So ATP gets broken down into ADP, that's adenosine diphosphate, because it's got 2 now, plus a lonely little phosphate there, okay, which we see there. Sometimes written as P, sometimes written as PI, which stands for inorganic phosphate. But yeah, that's the cycle that happens there. And what's interesting as well is this relates back to our enzymes topic because we've got an example of a degradation reaction here, and we've also got an example of a synthesis reaction here. So this is great because we have both synthesis and degradation working side by side to both break down the ATP into ADP and then build the ADP back up into ATP. Okay, and this cycle just happens continuously throughout the body. We only have about 50 grams of ATP in our bodies, so we have to constantly cycle it so we can get that back, so we can generate enough energy for all of our cellular activities. And this here is how we typically see this examined in a question paper. And I'll show you this reaction here where ATP is broken down into ADP plus PI, but then ATP, ADP plus PI can be regenerated or built back up into ATP. Okay, so that process just happens cyclically like that. And it's worth remembering both that chemical name, ATP, and this one here, ADP plus PI, which is the product when ATP is broken down. If you can remember that, it should help you out. There are questions that come up in the exam about that. And lastly, the energy transferred by ATP can be used for cellular activities. And some of those cellular activities include muscle cell contraction, the contraction of our muscle cells. We can see a wee diagram of that here. Okay. Cell division, which we'll learn a little bit more about at the start of Unit 2. Protein synthesis, which we learned about in topic 1.3. and the transmission of nerve impulses, okay? And again, we'll learn more about electrical impulses in Unit 2, but that's again an important one. For example, simply to move, to send a signal to move your body involves the transmission of nerve impulses. To think involves the transmission of nerve impulses. For your heart to receive the signal to beat involves the transmission of nerve impulses. It's a super important activity, and we can only do that because of this molecule here, this ATP, okay? And that ATP is generated from the breakdown of glucose, okay? And it's the chemical energy that's stored in the glucose that is released and allows us to do that. And this process, this whole process, is called respiration, and it's controlled by enzymes, folks. So that's the first thing that we're needing to learn about here. Now, there are actually a couple of ways in which the reaction can go in terms of depending on the conditions that the cell is in at that particular time. So the first one we're going to talk about is something called aerobic respiration. Now, aerobic respiration takes place in the presence of oxygen. Now, respiration will always involve the breakdown of glucose. Okay, glucose will always be broken down. And when it's broken down, so two molecules of pyruvate are released from one molecule of glucose being broke down. And we also release enough energy to get two molecules of ATP. So two molecules of ATP, two molecules of pyruvate, all the twos. Glucose broken down into two pyruvates and two ATPs. Now, this reaction over here, that will happen pretty much regardless of the conditions that the cell is in. So you'll always be able to make a tiny amount of energy, two ATP, two molecules of ATP, which is a relatively small amount. The further breakdown of pyruvate depends on the presence or absence of oxygen, okay? So if the oxygen is present, then we start the process known as aerobic respiration. Aerobic means with oxygen. So aerobic respiration will take place if oxygen is present. We can see that here in this diagram. When the oxygen is present, pyruvate can then be broken down into the products carbon dioxide, water, and it produces a large number of ATP molecules, okay? A large number of ATP molecules. And we also get these products here as well, carbon dioxide and water. That's just because glucose, which is the molecule that's initially broken down, is full of carbons, hydrogens, and oxygens. So as a result, our waste products are carbon dioxide, CO2, and water, H2O. Okay, but the main thing that we're getting out of this is the large number of ATP molecules. That is the key here. Okay, so when oxygen is present, our cells will undergo aerobic respiration and produce a much greater number of ATP molecules, a large number of ATP molecules. Now, the question I get asked most often from students is, what is that number? Now, we used to learn the number and we would use it for the exam. It could be examined, but now they just simply want you to say a large number. And not to confuse you, but usually an additional 36 on average molecules are made in this process, which gives us a total of 38 when we add in the two that were made from that first reaction. Okay, and again, I say usually because sometimes we don't produce the full 38 ATP for each one molecule of glucose that we break down. It varies. So that's why I think we now just say a large number of ATP molecules are made. So this is aerobic respiration. In the presence of oxygen, aerobic respiration takes place. Now, you'll remember from unit topic 1.1 that aerobic respiration takes place in the mitochondria. This first reaction up here actually takes place in the cytoplasm, and then it's this point here where it moves to the mitochondria, the second set of reactions here. Okay, so hopefully that makes sense. That's aerobic respiration, takes place in the presence of oxygen. When oxygen is absent, okay, when there is an absence of oxygen, something called the fermentation pathway will take place. Now, fermentation pathway in animal cells, so for example, humans, okay, in our cells, that absence of oxygen will mean that we won't produce water, carbon dioxide, and a large number of ATP. Instead, as animals, we will produce a substance known as lactate, okay? And if we were plants and yeast, we would produce a set of waste products, carbon dioxide and ethanol. Okay. Now, fermentation is not a ideal energy pathway. If we use fermentation for too long a period, we're going to run out of ATP. We're not going to have enough ATP to sustain the complex multi-celled organism that we are with all of the cellular activities that are taking place. And that's why, for example, humans can't survive without much oxygen for a very long period of time. Okay, so you'll know yourself. I guess at PE you might have had to do 200 or 400 meters or whatever for the sports day or sports day heats. And as you're running them, you realize about halfway around you cannot continue to run at the pace you're running at because your muscles are screaming at you. You cannot sustain your top maximal output of power, your top maximal output of energy. You can't sustain that because your cells are unable to get enough oxygen in to maintain aerobic respiration. So at that point, fermentation starts to take place. And the issue with fermentation is, of course, that it releases fewer molecules. It only releases 2 molecules of ATP compared to the large number in aerobic. And that's what eventually causes you to slow down and not able to sustain your top output, your top pace the whole time while you're running. So this process, while useful, is not ideal. Ideally, have oxygen present and undergo full aerobic respiration. But in the absence of oxygen, the fermentation pathway will still take place. And for animal cells, that produces lactate. Okay, and you might have heard of something called lactic acid, which does accumulate in your muscles and your blood, and it can cause pain and fatigue within that area of the body. And plants and yeast produce carbon dioxide and ethanol. And you might have heard of fermentation in terms of producing things like alcohol and so on. That uses that method because ethanol is a byproduct, which is obviously the alcohol part of alcoholic drinks like wine and beer and so on. Okay, so that's in the absence of oxygen. The fermentation pathway takes place. Animals produce lactate, plants and yeast produce carbon dioxide and ethanol. And this whole process produces only the initial molecules that were made in the first breakdown, which are 2 ATP. Now, I mentioned it earlier on, but respiration begins in the cytoplasm. So this initial breakdown of glucose to pyruvate, that will happen in the cytoplasm. Okay, reaction 1, glucose to pyruvate to 2 molecules of pyruvate, I should say, and 2 ATP. That happens within the cytoplasm. Now, if oxygen is present, then this reaction continues on to the mitochondria and aerobic respiration will take place only if oxygen is present. If oxygen is not present, then fermentation pathway will take place and that will occur within the cytoplasm. Okay, but aerobic respiration will go on to take place in the mitochondria. So you need to know the location of each of these pathways: fermentation, cytoplasm, aerobic respiration, mitochondria. And the good news is you should probably be remembering that from topic 1.1 anyway, that the mitochondria is the site of aerobic respiration. Now, the last thing to note about respiration is this. Because a larger number of ATP are produced when aerobic respiration takes place, the higher the energy requirement of a cell, the greater the number of mitochondria will be present in that cell. So, for example, we talked about muscle cell contraction earlier on requiring a large number of ATP, right? It's a cellular activity that needs a lot of ATP. So therefore, in order to produce the number of ATP needed for the muscles to contract all day long as you're walking around and doing things, and maybe taking part in physical activity, we need a larger number of mitochondria present within that cell. And if we look at the diagrams here, we can see the muscle cell has, I mean, this is not the actual numbers, it's just to represent it, but we've got 6 mitochondria here in the muscle cell, and something like a skin cell, which has a lower energy requirement, not no energy, but lower. That only has the 1 mitochondria by comparison there. Okay. So some cells have a higher energy requirement and therefore require a greater number of mitochondria. And again, that's a question that can be asked in the exam. It's like, why does X cell have more mitochondria than Y cell? And you simply say because X cell has a greater energy requirement. And if you know what the cell does, you can say what it needs that energy for. So if it's a muscle cell, it's for muscle contraction. If it's a neuron, it's for the transmission of nerve impulses. If it was a sperm cell, it maybe be energy to swim. Okay, to move its tail to swim. Something along those lines would be a suitable answer for that. And the last wee bit about respiration that you need to know about is about the equipment that we use to measure the rate of respiration. So we use something called a respirometer. Okay, a respirometer. Meter meaning to measure, and respiro obviously for respiration. So respirometer measures the rate of respiration in organisms. Okay, so how quickly respiration is happening within that organism. So, for example, we've set up a respirometer here. Okay, this is just a diagram from a past paper, and it's set up to measure the effect of temperature on the rate of respiration in germinating or growing peas. Okay, so we can see we've got the peas here. They're starting to germinate. Respiration will be happening. It needs energy to help it germinate, so respiration will be occurring within the cells here. And we've got a thermometer which tells us the temperature. And what we're going to do is we're going to vary the temperature that these peas are exposed to and see how that affects its rate of respiration. Now, how do we measure the rate of respiration in peas? Well, we know one of the products of respiration is the production of carbon dioxide. So what we can do is we can measure how much the liquid level in this particular one is moving with the amount of gas that is released by this particular germinating pea. Okay. Quite often we use a gas syringe, for example, to capture that gas that's released from it. There are lots of ways to measure it, but essentially we are measuring the products that are made by these peas, whether that be the carbon dioxide, which will be measured in a variety of ways, or some other measurement. Okay. So the effect of temperature and the rate of respiration in germinating peas. Now, what you should be able to do for your exam is you should be able to describe a suitable control experiment, okay, for this particular experiment up here. Now, a control, I always teach my classes COCO, so control is a comparison, okay? Now, what are we comparing? Because you can't just say it's a comparison. What we're comparing is both with and without the thing that we're investigating. Now, what we're investigating here is the rate of respiration in germinating peas. So, in a control experiment, we would be removing the thing we're investigating, which is the germinating peas. Now, in an experiment, we're only allowed to change one thing, and we're already changing the temperature. So, therefore, if we are going to replace these germinating peas, we have to replace it with something very similar. And the most common thing we do in these experiments is we either put in some glass beads that are roughly the same size and mass as the germinating peas, or we can boil the peas. And when we boil the peas, we denature the enzymes, which means that respiration cannot take place in the peas. Okay, so we don't have germinating peas anymore. We have boiled peas, which will not germinate because all its enzymes have been denatured by the boiling process. Okay, so we can replace those peas either with glass beads of equivalent size and so on, or we can replace them with boiled peas because the enzymes are all denatured and therefore respiration cannot take place. Okay, so that's a little skills bit there just to add on to the content knowledge of this particular topic. So I hope that was useful. As always, leave a comment to let me know if you enjoyed the lesson, if there's things that clarified for you, or if there's things you're still unsure about. What we're going to do next is we're going to move on to the practice paper questions. Okay, folks, so welcome to the second part of the lesson where we look at some past paper questions to see how we can apply our knowledge to the topic of respiration. Question one here. Question one's actually a skills question, but we'll do it anyway because it's good to sort of learn about these skills as we go along and integrate it into the context of what we're learning. So it says the diagram shows an experiment which can be used to find the energy content of different foods. Each food was completely burned and the energy content was estimated by the rise in temperature of the water. Okay, so the reliability of this experiment could be improved by. Now, I always teach my students RIRI, so reliability repeat. Okay, if you remember RIRI, not Rihanna, but RIRI in terms of the scientific context, it is reliability repeat. Okay, so we're ideally looking for the word repeat or something which suggests that this experiment has been repeated. So let's have a look. Burning each food for the same length of time. Okay, that would help with validity because you're keeping that variable the same, but it won't help with reliability. Repeating the experiment with each food several times. Now that's obviously got repeat in it and it's doing the experiment with each food. So we're doing all of the foods again several times. So that sounds pretty good to me, but we'll check the other ones anyway. C, removing the thermometer from the tube to read it accurately. Accuracy is not really to do with reliability, so that's not going to be one we're looking for there either. And lastly, repeating the experiment. So repeating the experiment, that's good. Using a different food each time. Okay. So what they're suggesting there is that they are going to change the food every time they repeat the experiment. And that's not really what we're after. What we're ideally after is to repeat the experiment with each food several times, because what that allows you to do is to get several results for each food and calculate an average, okay? And that average will probably be closer to the true mean than if we only did it once, okay? So we improve the reliability by repeating the experiment for each food several times there. Okay, number two, the apparatus shown was used to investigate the rate of respiration in yeast at 20 degrees. Which of the following changes would cause a decrease in the rate of respiration of the yeast? Right. So we're dealing with respiration here and how quickly it goes. Now, what are the things that respiration need? That's where my head would be at straight away. What does respiration need? Well, respiration needs to begin with glucose. Okay, now we have glucose in this experiment. We have a 5% glucose solution that's in with the yeast. So if we wanted to decrease the rate of respiration, in my head, we would decrease the amount of glucose that we give to the yeast. Okay, I'm not sure if that's going to be an answer, but that's where my head is at before I even get to the options. Okay, all of these other things are obviously part of the experiment, but probably are less of a factor in terms of the rate of respiration. So if we look at A, we increase the thickness of the layer of oil by 1 millimeter. Well, I don't know if that'll make much of a difference. It's already relatively thick there. I can't see any issues in terms of that. In terms of it, that's supposed to be there to keep the air in there and to ensure that fermentation continues to happen. But, there's no oxygen getting in. But like again, the thickness of that I don't think is going to make any real difference here. B, increasing the temperature of the glucose solution by 1 degree. Now, if you increase the temperature of the solution, what are you actually going to do? Well, you're actually going to make the enzyme activity increase. You're going to make the enzyme more active. Okay, and now we know that respiration is controlled by enzymes. So if we make the enzymes more active, we're actually going to increase the rate of respiration there, not decrease it, which is what we want. Okay, so although that sounds like a good idea, it's actually going to do the opposite of what we want. C, decrease the concentration of the glucose solution by 1%. Well, that chimes with what we suggested earlier on about the glucose going down. So we'll keep that as a possibility and decrease the volume of indicator solution by 1 centimeter cubed. Again, that's not really going to have any significant effect there. Okay, less indicator solution. And that indicator solution is indicating the carbon dioxide that's released in this process. So, there'll be a colour change, but I don't think the volume of that will have any real. In fact, it won't have any impact on the rate of respiration. It just might have an impact on us measuring the CO2, but that's a different thing. So the answer we're looking for there is going to be C. Okay, question 3. There was 4 flasks, J, K, L, and M, and they're set up to investigate the production of carbon dioxide during respiration. Lime water turns increasingly cloudy as more carbon dioxide passes through it. Predict what would happen if only one worm was used in flask L, and this is what would happen. The lime water in the flask would. Okay, well, let's look at the experiment. So we've got 5 worms here. Okay, we've got 5. And we've got air that's flowing in one direction from this left-hand side to the right-hand side. What that means is that the air that's going into K here can only come from J. The air going into L can come from K, and the air going into M can come from L. Okay. Now, initially, we've got a sodium hydroxide there absorbing CO2. That's fine. So that would suggest there's no CO2 in the air that's going into here. We've got some lime water here. That would hopefully measure that there's no CO2 and it wouldn't turn cloudy. In L, we have all these earthworms. Now, these worms will be undergoing respiration and they will be producing carbon dioxide. So that carbon dioxide produced by the worms should go up through this tube here and down into this lime water. Now, the lime water turns increasingly cloudy as more carbon dioxide is passed through it. So I would guess that lime water is going to go cloudy pretty quickly because there are 5 worms here, all producing CO2. So let's have a look at what the options are. It says the lime water in flask K would turn more slowly. Okay, so K is here. Now, if we change that from 5 to 1 earthworms, will K be affected? No, because it comes earlier in the process and we know the air is only flowing in one direction. So it's not going to be that one. K would turn cloudy more quickly. Again, K is not going to be affected here, so we can eliminate that one. So M comes after the worms, so that will be affected. So M would turn cloudy more slowly. Well, lime water turns cloudy as more carbon dioxide is passed through it. With one worm instead of five, we've got one worm instead of the five, then I would imagine it would turn cloudy more slowly because less CO2 is released by one worm compared to five. So I'm thinking C is going to be our answer here. And if we just check for D, M would turn cloudy more quickly. Well, that's not likely to happen if you've only got one worm compared to five. If you said we're changing it from 5 worms to 10 worms, then D would be the answer. But because we are decreasing the number of worms and therefore decreasing the CO2 produced, it's going to be C here. Okay, question 4. Which of the following reactions takes place during fermentation in plant cells? Right. Plant cells are different from animal cells. In fermentation in animal cells, we produce lactate, and in fermentation in plant cells, we produce carbon dioxide and ethanol. Okay, and that's just something you have to memorize. So which one is it going to be? Well, it's not going to be D. That's lactate. It's not going to be this one because it produces water and ethanol. This one here produces carbon dioxide and water from glucose. That sounds good, but that's actually for aerobic respiration. We'll score that out. We are looking at fermentation, remember? And A is pyruvate to carbon dioxide and ethanol. So that's going to be our answer there, okay? Remember, glucose is broken down into pyruvate, and then the pyruvate is broken down into carbon dioxide and ethanol. Number 5, which of the following substances increases in concentration in contracting human muscle cells, okay? So we're thinking about animals here. When the respiration occurs in the absence of oxygen, okay? So animal cells, absence of oxygen. So we're thinking fermentation here. And fermentation in animals produces lactate, which is also known as lactic acid here, okay? So it's this one here we're looking for. If it said plant or yeast cell, then ethanol would have been the right answer, but because it's human or animal cells we're dealing with, it's lactic acid, okay? Now, you're probably wondering as well, why is it not ATP? ATP is produced in the previous step, the breakdown of glucose. So when there's an absence of oxygen in human muscle cells, they produce lactic acid or lactate. Okay, number 6, we've got an experiment set up here. What's this looking at? Investigating the rate of respiration in yeast. Okay. Which 2 experiments should be compared to show the effect of temperature on the rate of reaction? Now, in an experiment, if you are investigating temperature, that is the thing that you're going to change, and it's the only thing that you're going to change. Okay, if we're looking at the effect of temperature, we only change the temperature. So what it's asking us is which 2 experiments have only the temperature changed. Okay, which of the 2 experiments only have temperature changed and everything else is kept the same? So if we look through them, this one here is set up at 22, that's the same, and these 2 at 30, okay? So if we're comparing temperature, we're going to either go with one from the top and one from the bottom, okay? And we have to look at all the other factors as well. So for example, this is 1 gram yeast and 2% glucose. Now, do any of these down here have that? 1 gram yeast, 5%, that's not right. 2 grams yeast, 2%, that's not right either. So 1 does not match with either 3 or 4, okay? It also doesn't match with 2 in that regard, but they're at the same temperature anyway, so they wouldn't have gone together. If we look at experiment 2, which is at 22 degrees, and these 2 are at 30, so 2 probably goes with 3 or 4. We look, it's got 1 gram of yeast and 5% glucose. Do any of 3 or 4 match that? Well, number 3 also has 1 gram of yeast and 5% glucose. And the only difference between experiment 3 and experiment 2 is the temperature, okay? So our answer there is going to be 2 and 3, which is going to be C. All right. So if we're looking at the effect of temperature, that's the only thing that can change. Everything else must remain the same. All right. That's how we work that one out. Let's go into some section 2 questions just for a bit of change of pace. This is a good one, actually. Muscle tissue can be dark or light in colour. Dark tissues use oxygen to release energy. The light cells do not use oxygen to release energy. So immediately, before I even look at the questions, I'm thinking to myself, uses oxygen? That's aerobic respiration. Okay, in my head, that's what I'm thinking. And light tissues do not use oxygen. So immediately I'm thinking that's fermentation for that one. Okay, before I'm even getting to the questions. And that's a good process to have as you approach questions in the exam. Already be thinking ahead, okay, this information they've given me, why are they giving me it? Well, if they're mentioning oxygen in a respiration question, then it's probably because they want me to think aerobic respiration. And if they're saying do not use oxygen, they want me to think fermentation. So question A, name the process by which energy is released in dark tissue cells. Okay, so dark tissue cells use oxygen. So we've already answered this question because we thought ahead. It's aerobic respiration for that one. Okay, so 1A there, aerobic respiration. B part 1, name the substance which muscle cells break down to produce pyruvate. So the substance which our cells break down to produce pyruvate, again, this is just a knowledge question. That's going to be glucose. Okay, glucose for this one. B part 2, when pyruvate is being formed, enough energy is released to form two molecules of a high energy compound. Complete the word equation below to show how this compound is generated. Okay, so there's something plus something gives something else. Now, first of all, we're thinking a high energy compound. What compound or molecule do we have in the body that is high energy? And immediately my mind is going towards ATP. Okay. And then I'm thinking, well, how do we make ATP? And if we remember earlier on, there was a synthesis reaction where ADP, adenosine diphosphate, plus our little individual phosphate, they join together and they form ATP. Remember, ATP, 3 phosphates, ADP, sorry, 2 phosphates, and then there's 1 phosphate there. So 2 plus 1 gives you 3. Okay, that's one of the ways to think of it there. Adenosine triphosphate from ADP plus PI. Okay, question C there. The table shows the average percentage of dark and light tissue cells. These cells were found in the muscles of athletes training for different events at the 2014 Commonwealth Games in Scotland. Okay, perfect. Use information in the table. Identify which type of athlete would likely produce the most lactic acid in their muscle cells and justify your answer. Okay, so we need to go back to our information at the top for this one. So dark tissue cells use oxygen, aerobic respiration. Light tissue cells do not use oxygen, fermentation. So therefore, if we're looking at which one would produce the most lactic acid, that's going to be through the process of fermentation. Okay, that's what produces lactic acid, not aerobic respiration. So therefore, fermentation is related to our light tissue. So let's go down to our table here, and we're looking at light tissue. Okay, because that's the one involved in fermentation. And if it's the one that produces the most lactic acid, it's going to be the one that has the most light tissue cells, which is going to be here. And our answer is going to be the sprinter. Okay, so the type of athlete is the sprinter. And the justification is that they have the highest percentage of light tissue cells. The sprinter who has the highest percentage of light tissue cells. Part 2, a sample of muscle tissue from an athlete was examined and found to contain a total of 360 cells. 90 of these cells were light tissue cells. Identify the type of athlete the sample was taken from. Now, this doesn't have raw numbers in it here, it has percentages. So we are told that we just have 90 out of 360. So we have to work that out as a percentage, okay? So we do 90 over 360 and we times it by 100. Okay? And if my mental maths is right, 90 is a quarter of 360, so that should be 25%. Okay? So actually, the percentage is 25%. And when we go up to the table, we find 25% here. So this person that we're talking about is going to be the swimmer. Okay, so it's the swimmer. Number 2, the table shows information about the 2 types of respiration in animal cells. Tick the boxes in the table to indicate whether the statements apply to aerobic respiration, fermentation, or both. All right, and I should have said at the start, but just to remind you, if you do want to do these questions as we go, be sure to pause the video and do them and then come back to hear the answers and the explanations. Okay, so oxygen is required. Well, that is definitely aerobic, but it's not fermentation. Fermentation is the absence of oxygen, so we do not need that for that one. Pyruvate is formed. Well, yes, aerobic respiration, glucose to pyruvate, which then is converted into carbon dioxide, water, and a large number of ATP. With fermentation, again, pyruvate is formed, and the pyruvate becomes other things like lactate or carbon dioxide and ethanol, depending on the organism. Lactate is formed. That's only going to happen during fermentation in animal cells. Okay, it will not happen during aerobic respiration. And carbon dioxide is formed. Well, we know that aerobic respiration produces water, carbon dioxide, and a large number of ATP, so that one there. But we also know that fermentation in plant and yeast cells produces carbon dioxide and ethanol. Okay, so we're looking to tick that box too. Okay, B, ATP is an energy-rich molecule formed by respiration. Name the cellular process which requires energy. Name a cellular process, which requires energy from ATP. So a cellular process. Now, we looked at four of them on our slides when we were doing the lesson. One of them involved the muscular contractions, the movement of our muscles. One of them involved cell division. There was another one for protein synthesis, and the last one was the transmission of nerve impulses. So you can choose any of those four because it only asks for one, and write it down here. Okay, so muscle contraction, transmission of nerve impulses, protein synthesis, and cell division are your four options there. Okay, I think we'll leave it there because that's us going through quite a few questions. But as always, leave me a comment if you want to ask more about anything else, if you want to clarify anything I've said, or if you need any more help on any particular area, you can recommend that to me. Okay, folks, that's all for now. Class dismissed.